A man is lying dead on a metal table in Berlin, and the pathologist leaning over him cannot smell the thing that killed him. Everyone else in the room can. They keep mentioning it, almost apologetically, the way you'd mention a smell in someone else's kitchen. Bitter almonds. Sharp. Unmistakable, they say. The pathologist nods, writes it down as a second-hand fact, and gets back to work. Because for him, the room smells like nothing. Formaldehyde, maybe. Cold air. Nothing almond-shaped at all. This is not a failure of training. It is not a bad day. It is genetics doing something almost mischievous, quietly withholding one specific piece of sensory information from roughly 4 in every 10 people on the planet, including, occasionally, the very professionals whose job is to notice it. And once you learn that fact, a strange question starts nagging at you. How much of forensic history, how many verdicts, how many quiet dismissals of natural causes, how many poisoners who simply weren't caught hinged on whether the particular doctor in the particular room that particular day happened to have the right gene? That's the story, not just the chemistry of a poison, the much stranger story of how an entire branch of human knowledge, crime detection, was built for centuries on a signal that a huge percentage of the species literally cannot receive. Cyanide's reputation precedes it. Ask almost anyone in almost any country whether they know what cyanide smells like, and they'll answer immediately: bitter almonds. It's one of those facts that seems to belong to common sense itself, taught in mystery novels, crime shows, chemistry classes, half remembered from 100 detective stories where the clever inspector leans over the body, sniffs, and announces the cause of death before the coroner has even opened his bag. Here's the part nobody puts in the mystery novel. The inspector sniffing that corpse has, statistically, something like a 40% chance of smelling absolutely nothing. The ability to detect hydrogen cyanide by smell is not a universal human trait. It is a specific genetic sensitivity tied to particular olfactory receptors, and it is inherited the way eye color or the ability to roll your tongue is inherited. Some people have it, some people categorically do not, and there is nothing about intelligence, training, or effort that changes that fact. You cannot practice your way into smelling cyanide. You either carry the genetic machinery or you don't. This single detail, buried in toxicology textbooks, occasionally mentioned in forensic training, rarely discussed anywhere else, quietly rewrites the entire mythology of the poison with the telltale smell. For as long as cyanide has been used, feared, and investigated, a huge fraction of the people investigating it have been working the case with one sense effectively switched off. Which raises an obvious question, the kind the narrator of a documentary like this can't resist asking. If the signature smell of the most famous poison in history is invisible to nearly half of the population, what does that mean for every case where someone swore they smelled nothing unusual at all? Before going further into the genetics, it's worth being precise about the substance itself, because cyanide gets used loosely in fiction as a catch-all word for instantly fatal poison, and the real chemistry is both simpler and stranger than the legend. Cyanide refers to a compound containing a carbon atom triple bonded to a nitrogen atom, the cyanide ion. In its most infamous forms, it appears as hydrogen cyanide gas, or as solid salts like potassium cyanide or sodium cyanide, both of which release hydrogen cyanide gas when they contact acid, including the acid naturally present in a human stomach. What makes cyanide so lethal isn't that it attacks like a blade or burns like a corrosive. It works on a cellular level, on a mechanism found in essentially every cell in the body: cellular respiration. Mitochondria, the structures that convert oxygen and nutrients into usable energy, depend on an enzyme called cytochrome C oxidase to complete the final step of that process. Cyanide binds to this enzyme and blocks it almost completely. The result is a body full of oxygen that its own cells can no longer use. Victims don't suffocate from lack of oxygen in the blood. They suffocate with oxygen-rich blood that their tissues simply cannot metabolize. It is, in a very literal sense, death by a locked door rather than an empty room. This is why cyanide poisoning can kill within minutes at high enough doses, because it isn't waiting for a slow chemical cascade. It's switching off a machine that every cell in your body depends on within seconds of exposure. And this is also why the almond smell, when a person can detect it at all, became such a celebrated clue. Hydrogen cyanide has a genuinely peculiar odor profile, faintly sweet, sharp, and yes, reminiscent of bitter almonds. Bitter almonds themselves contain a related compound, amygdalin, which breaks down into hydrogen cyanide, which is why raw bitter almonds, in enough quantity, are mildly toxic in their own right. To someone who can detect it, it's often described as immediate and distinctive. To someone who can't, there's nothing there at all. Not a faint smell, not an ambiguous chemical whiff, nothing, just ordinary air. Fast fact: the concentration at which a sensitive person can detect hydrogen cyanide by smell is roughly in the same range as concentrations that can already be dangerous with prolonged exposure, meaning even for people who can smell it, the warning often arrives close to the moment the danger begins, not safely before it. The ability itself has a name in the scientific literature: Cyanide Anosmia, a specific inability to smell hydrogen cyanide while retaining normal smell for essentially everything else. Studies conducted across the 20th century, some of the earliest published in the 1930s, refined repeatedly through later decades, consistently found that somewhere between a quarter and just under half of tested populations could not detect it at typical concentrations, with many toxicology and forensic references settling on a commonly cited figure of around 40%. It's inherited, and researchers have long treated it as behaving like a recessive trait tied to specific olfactory receptor genes, meaning two people with normal smell in every other respect can, purely by the luck of which combination of genes they inherited from each parent, land on opposite sides of this particular sensory divide. One sibling smells the almond note instantly. Another sibling, raised in the same house, eating the same food, would stand in a room full of the gas and swear it smelled like nothing.
Here's where the narrator's favorite kind of question shows up. If this is such basic, testable biology, if it's been documented in scientific journals since before the Second World War, why isn't it common knowledge? Why does virtually every crime drama still treat the bitter almond smell as a universally reliable clue, as though every detective, every witness, and every coroner in history had identical noses? Part of the answer is simply that fiction prefers clean rules to messy biology. The smell always gives it away is a satisfying story mechanic. The smell gives it away to some people some of the time, unpredictably, depending on genes nobody in the room has been tested for, is accurate, but it ruins the pacing of a whodunit. The other part of the answer is more uncomfortable. Real forensic history has, on more than one occasion, actually run into this exact problem in cases where lives, verdicts, or reputations depended on somebody's nose. Myth 1. Question: can anyone with a normal sense of smell detect cyanide poisoning simply by being in the room? Verdict: false. Reality: cyanide anosmia is a specific, well-documented genetic condition separate from general smell ability. A person can have a perfectly ordinary, healthy sense of smell for coffee, perfume, garbage, and everything else in daily life, and still be completely unable to detect hydrogen cyanide because the receptors involved are specific to that compound. Why the myth became popular: detective fiction going back well over a century needed a clean, dramatic sensory clue that a clever investigator could notice and an ordinary witness could miss due to inattention rather than biology. The smell was there, you just weren't paying attention is a much better story beat than the smell was there, but you are one of the roughly 2 in 5 people who cannot physically perceive it. Evidence: peer-reviewed olfactory genetics research spanning from early 20th century smell testing studies through modern receptor gene research consistently finds a substantial minority to near majority of any tested population unable to detect the compound, regardless of general smell health. Cyanide compounds have been part of human chemistry for a very long time. Prussian blue, the pigment from which the modern understanding of the cyanide ion partly emerged, was discovered by accident in the early 1700s by a color maker in Berlin trying to synthesize a red dye and instead producing a deep, stable blue that would later reveal a whole family of related compounds. From that pigment chemistry, over the following century, scientists gradually isolated and understood hydrogen cyanide and its salts, and by the 1800s, cyanide compounds had entered both industry and, inevitably, crime. The 19th century was something of a golden age for poisoners in Western Europe and the United States, not because people were more murderous, but because forensic science hadn't yet caught up to chemistry. Arsenic dominated the era's poisoning cases in part because it was cheap, available in household products, and, crucially, essentially undetectable in a body using the tools available before the 1830s. Cyanide occupied a smaller but persistent niche, harder to obtain in some periods, easier in others depending on industrial regulation, and carrying that supposedly unmistakable smell that many popular accounts assumed would betray any poisoner instantly. This is where the genetics problem becomes more than trivia. Coroners, physicians, and early forensic investigators of the 1800s had no concept of cyanide anosmia as a defined trait. The scientific literature establishing it as a specific genetic phenomenon wouldn't really solidify until the 20th century. So, for the entire span of cyanide's use as both an industrial chemical and an occasional murder weapon throughout the 1800s, any given examiner's ability to detect it by smell was essentially a coin flip nobody knew they were flipping. Multiple historical accounts of 19th century and early 20th century poisoning investigations describe physicians and even morticians who reported detecting no unusual smell at a scene, later confirmed through chemical testing to involve cyanide, not because they were careless, but very likely because of exactly this kind of sensory blind spot. The historical record can't always distinguish between no smell was present and no smell was perceived, which means we will likely never know precisely how many early cases were miscategorized simply because the wrong nose happened to be in the room. What's known, cyanide anosmia is real, genetically based, and affects a substantial share of the population, commonly cited around 40%, though estimates in different studies range somewhat depending on methodology and population sampled. What's likely, some number of historical poisoning deaths, particularly before reliable chemical testing existed, were misattributed to natural causes, partly because examiners could not smell the evidence in front of them. What's still debated, exactly how many cases this affected and to what degree, is not something historians or toxicologists can quantify precisely, because by definition, the cases most affected are the ones where nobody suspected poisoning at all, and therefore no records exist to review. This is less a gap in the research and more a structural blind spot in the historical record itself, the ultimate example of we don't know what we don't know. The reassuring part of this story is that human ingenuity, as usual, eventually built a workaround for human biology's inconsistency. Chemistry didn't need everyone to have a good nose. It needed reliable tests that worked regardless of who was standing in the room. Early chemical tests for cyanide date back to the 1800s, refined across decades by chemists working specifically because they knew smell alone was an unreliable method, though the specific genetic reason wouldn't be documented for a long while yet. Chemists observed early on that smell tests produced inconsistent results between examiners, which was reason enough to develop something more rigorous.
One of the more elegant historical solutions was the Prussian blue test, which leaned on the same chemistry that had accidentally produced that famous pigment a century earlier. Exposing a sample suspected of containing cyanide to iron compounds under the right conditions produces a visible blue color, a chemical reaction that doesn't care whether the chemist doing the test can smell almonds or not.
Later, more sophisticated methods, colorimetric tests, and eventually gas chromatography and mass spectrometry in the 20th century moved forensic toxicology almost entirely away from sensory judgment and into instrumentation, precisely because instruments don't inherit genes. This is, in miniature, one of the great underappreciated stories of forensic science generally. The field's credibility didn't come from sharper senses, braver investigators, or more dramatic deductions. It came from removing the human sensory system from the loop as much as possible, replacing "I smelled something odd" with "the sample produced this measurable reaction," because measurable reactions don't vary by whoever happens to be holding the test tube. There's an economic and institutional angle worth pausing on here. Chemical testing methods were expensive and slow to adopt in many jurisdictions well into the 20th century, particularly outside major cities, which meant that in poorer or more rural regions, sensory judgment by an attending physician often remained the practical, if unreliable, first line of suspicion for decades after better methods existed elsewhere. Resource allocation, not just scientific ignorance, shaped how long the sniff test era persisted in different parts of the world. Cyanide's cultural reputation as a dramatic, almost cinematic poison owes a handful of historically significant episodes, and it's worth walking through a few of them with this genetic wrinkle in mind. During the Second World War, cyanide compounds were used on a horrific industrial scale, and the substance's association with mass atrocity is one of the darkest chapters connected to this chemistry, a somber historical fact that sits well outside the realm of any clever detective narrative and deserves to be acknowledged as such, rather than treated as documentary trivia. In criminal poisoning cases across the 20th century, cyanide appeared periodically as a weapon in high-profile murders and suicides, and forensic reports from several of these cases explicitly noted that investigators or witnesses at the scene did not detect any unusual odor, details that, read today, line up suspiciously well with what we now know about the prevalence of cyanide anosmia, even though the reports themselves, written before the trait was well understood, simply noted the absence of smell as an observation, rather than explaining why. Perhaps the most infamous single incident involving cyanide in American memory is the mass deaths at Jonestown in 1978, where a cyanide-laced drink was used in a mass murder-suicide of over 900 people. Survivor and investigator accounts from the aftermath described widely varying sensory experiences among those who came into contact with the aftermath, some reporting a distinct smell, others reporting none at all, a pattern entirely consistent with a population containing both cyanide smellers and cyanide anosmics working the same horrific scene. None of this is meant to treat tragedy as a puzzle to be solved for entertainment. It's meant to illustrate something genuinely important. Across radically different contexts, courtroom murder cases, wartime atrocity, mass tragedy, the same quiet biological inconsistency shows up again and again in the historical record, almost like a fingerprint of its own. It's worth pausing on a figure documentaries usually skip entirely, the small-town coroner or physician of the 1800s and early 1900s, untrained in chemistry, working without a laboratory, expected to make a snap judgment about cause of death using nothing but experience and their own senses. This person is not incompetent. They are doing a genuinely difficult job with the tools their era gave them, tools that we now know were quietly, invisibly unreliable for a huge share of the population performing the job. It's easy, watching history from a distance, to imagine forensic failures as failures of diligence or intelligence. Far more often, they were failures of information that didn't yet exist. The coroner in 1850 examining a body and smelling nothing unusual wasn't being careless. He may simply have been born without the genetic machinery to smell the one thing that would have told him everything. Fast fact: The gene variants associated with cyanide anosmia are believed to affect specific olfactory receptors, and researchers have noted that detection ability can also vary somewhat with concentration and individual sensitivity among people who generally can smell it, meaning the trait isn't simply a strict on-off switch for everyone, but for a very large minority it functions as exactly that, entirely off. Step back from cyanide specifically, and this story becomes an example of something much bigger, the assumption built into so much of scientific and forensic history that human senses are reliable, standardized instruments. They are not. Taste, smell, color perception, even pain sensitivity vary meaningfully across the human population due to genetics, and forensic history is full of quiet moments where an investigation's outcome depended on which particular human happened to be doing the sensing. Consider an adjacent, better-known example. Colorblindness, present in a notable percentage of men, has occasionally been relevant in historical contexts, requiring color judgment, signal reading, uniform identification, even some early forensic evidence involving stains and dyes. Consider supertasters and non-tasters, whose ability to detect bitterness in certain compounds varies by genetics tied to a specific taste receptor gene, a trait studied extensively in the 20th century that similarly shows humans are not standardized sensory instruments. Cyanide anosmia belongs to this same family of invisible variation, differences so ordinary and so evenly distributed throughout history that nobody thought to interrogate them until genetics gave researchers the tools to actually measure what people could and couldn't perceive, rather than simply assuming perception was universal. Today, industrial safety standards for facilities that use hydrogen cyanide gas, including certain mining, electroplating, and chemical manufacturing operations, do not rely on workers' smell perception at all.
Modern occupational safety protocols mandate electronic gas detectors and monitoring equipment specifically because regulators, informed by exactly this genetic research, understand that a meaningful share of any workforce simply cannot be trusted to smell a leak before it becomes dangerous. The lesson learned from decades of forensic and toxicological research about unreliable human noses has been quietly built into modern workplace safety law, even though almost nobody outside the field is aware that's where the rule came from. Myth 2. Question: was the Prussian blue chemical test named after the same chemistry as the paint color for a coincidental, unrelated reason? Verdict: false. It's the same underlying chemistry. Reality: the pigment, Prussian blue, and the chemical basis of certain classic cyanide detection tests are directly related, both arising from the interaction of iron compounds with cyanide-containing compounds. The accidental discovery of the pigment in the early 1700s predates its forensic application by roughly a century, but the same core chemical reaction underlies both. Why the myth became popular? The connection between a household paint color and a forensic poison test sounds too tidy to be true, so many retellings either omit it entirely or assume it's an invented flourish rather than genuine chemical history. Evidence: documented histories of both pigment chemistry and early forensic toxicology confirm the shared chemical basis, tracing directly back to the same class of iron cyanide compounds. This is the detail that tends to genuinely surprise people, so it's worth stating plainly. Yes, modern forensic pathologists, despite years of medical training, are not exempt from ordinary human genetics. A pathologist performing an autopsy today can be every bit as unable to smell hydrogen cyanide as anyone else with the relevant genetic variant, and forensic training programs and toxicology references have, for decades now, explicitly flagged this as a reason not to rely on smell as a diagnostic method, precisely because relying on any individual examiner's nose introduces a roughly coin flip level of unreliability into a process that's supposed to be rigorous. This is why modern forensic protocol for suspected cyanide poisoning never rests on anyone's sense of smell as primary evidence. Blood and tissue samples are tested chemically through colorimetric assays and more definitively through gas chromatography-mass spectrometry, which can detect and quantify cyanide levels with a precision no human nose could match, even in someone with perfect olfactory sensitivity. The entire chain of custody for a cyanide poisoning case today is built to be nose-independent, from the initial suspicion through to courtroom evidence, a system explicitly designed around the assumption that any given human examiner might be one of the roughly 40% who would perceive nothing at all, which introduces a huge gambling element. What's known? Modern forensic toxicology relies on instrumental chemical analysis, not sensory detection, for confirming cyanide poisoning in legal and medical contexts. What's likely? Historical reliance on smell as a diagnostic clue, common in earlier eras of forensic medicine and still present in popular fiction, produced an unknown but almost certainly non-zero number of missed or delayed diagnoses. What's still debated, there is no comprehensive historical audit of exactly how often this specific factor changed the outcome of an investigation, since it would require re-examining case files from eras with far less rigorous record-keeping than exists today. This remains an area more of informed inference than hard statistical certainty. There's a deeper, almost philosophical layer buried in this story, and it's one the narrator can't resist poking at. Humans are remarkably bad at recognizing the limits of their own perception. If you can smell cyanide, it seems self-evidently obvious that everyone can. The smell is right there, unmistakable, impossible to miss. If you can't smell it, you likely have no idea there's anything to miss at all. The room simply smells normal, full stop, with no internal signal suggesting anything is absent. This asymmetry, where the people with the sensory ability have no reason to doubt its universality, and the people without it have no way of knowing what they're missing, shows up constantly throughout the history of science, not just in this one case. It took carefully designed comparative testing, testing multiple people against the same stimulus and comparing results, to even reveal that the variation existed at all. Nobody figures out they can't smell cyanide by introspection. They figure it out only when someone else in the room insists there's a smell, and there manifestly, personally, isn't one. This is a small but genuine lesson in epistemic humility that historians of science return to again and again. So much of what any individual assumes is obviously true for everyone turns out to be true only for the specific biological hand they were personally dealt. Not every cyanide compound produces a strong smell even for people who can generally detect hydrogen cyanide. Solid cyanide salts in low concentration or cyanide dissolved in certain liquids may release far less gaseous hydrogen cyanide into the air than popular depictions suggest, meaning even a fully smell-capable investigator might detect nothing simply because too little gas has been released to reach a perceptible concentration. A second, entirely separate reason the unmistakable smell trope oversells what actually happens at most real scenes. It's worth asking, in the spirit of taking absurd questions seriously until they aren't absurd anymore, why has bitter almonds survived as a reliable fictional clue for well over a century, despite this genetic wrinkle being documented in scientific literature since the 1930s? Partly, it's narrative economy. A detective sniffing a room and instantly identifying a poison is a compact, visual, and dramatically satisfying beat that doesn't require the audience to understand toxicology. Partly, it's because the trope predates widespread public understanding of olfactory genetics, the unmistakable smell idea calcified into cultural common sense well before the science that complicates it reached popular awareness. And partly, it's simple survivorship bias in storytelling. Nobody writes a mystery novel where the detective sniffs the room, smells nothing, and the case goes cold for entirely biological reasons that have nothing to do with cleverness, because that's a less satisfying story, even though it may be the more historically accurate one. Fast fact: Some animals, notably certain birds, appear to be able to detect cyanide-related compounds at levels well below human perception thresholds, and canaries were historically used in mining precisely because of a broader sensitivity to toxic gases, though the famous canary in a coal mine tradition was primarily about detecting low oxygen and other gases like carbon monoxide, rather than cyanide specifically, a common point of confusion in popular retellings of that history. Myth 3. Question: does everyone who can smell cyanide describe it the same way? Verdict: not entirely. False as a strict claim. Reality: even among people who can detect hydrogen cyanide, descriptions of the smell vary. Some describe it as strongly almond-like. Others describe something closer to a musty or acrid sharpness, with only a faint almond quality. This adds yet another layer of unreliability to eyewitness or examiner testimony about smell in historical cases. Not only did roughly 40% of examiners potentially smell nothing, but among the remainder, descriptions of what they did smell weren't perfectly uniform either. Why the myth became popular? A single vivid, universally agreed upon descriptor is much easier to teach, remember, and dramatize than a spectrum of subjective sensory reports. Evidence: toxicology literature and forensic training materials have long noted variability in odor description among cyanide-sensitive individuals, alongside the separate and better-known variability in who can detect it at all. Zoom out far enough, and cyanide anosmia becomes one small case study in a pattern that runs through the entire history of forensic science, medicine, and even law. Institutions gradually learning, often the hard way, that individual human perception is not a universal standard, and building systems specifically designed to route around that unreliability. The move from the doctor's judgment to the standardized chemical test mirrors similar transitions across many fields, from eyewitness identification giving way to fingerprint and DNA analysis, from subjective symptom description giving way to blood tests and imaging, from a single expert's opinion giving way to peer-reviewed, replicated, instrument-verified findings. In each case, the animating realization was the same. Individual humans vary in ways they themselves usually can't detect, and any system that depends entirely on one person's unverified sensory report is a system with an invisible failure rate built into its foundation. Cyanide anosmia is, in a sense, a perfect miniature of this larger story, small enough to explain in a few sentences, but carrying the same lesson as far larger institutional reforms in medicine and law. Reliability comes not from trusting any one person's senses harder, but from building processes that don't require any single person's senses to be trustworthy at all. Myth 4. Question: is cyanide poisoning always fast and dramatic as depicted in film? Collapsed within seconds, foaming, instant death? Partially true, heavily exaggerated. Reality: at sufficiently high doses, cyanide can act very quickly, sometimes within minutes, and can produce distressing symptoms, including rapid breathing, confusion, and loss of consciousness. But at lower doses, or with certain routes of exposure, the progression can be considerably slower, sometimes unfolding over hours, and doesn't always present with the theatrical symptoms popular film has trained audiences to expect. Some poisoning cases historically went unrecognized specifically because the presentation was slower and more ambiguous than the dramatic version everyone expected to see. Why the myth became popular? Dramatic rapid death makes for compelling cinema and tidy plotting. A slower, more ambiguous medical presentation does not translate as cleanly to a 2-hour thriller. Evidence: clinical toxicology literature documents a range of presentations, depending on dose, exposure route, and individual physiology, contradicting the idea of one universal instantaneous symptom pattern. In the spirit of scientific honesty this kind of documentary owes its audience, it's worth being explicit about the edges of current knowledge. Researchers have a reasonably solid grasp of the prevalence of cyanide anosmia in tested populations, and a reasonably solid understanding that it's genetically inherited, tied to specific olfactory receptor variation.
What remains less certain, and likely always will, is the precise historical toll of this trait. Exactly how many poisoning deaths across the 18th, 19th, and early 20th centuries were misattributed to natural causes, specifically because of an examiner's genetic inability to smell the evidence. That number cannot be recovered. It lives in a category historians sometimes call the dark figure, an unknowable quantity of undetected events, structurally invisible precisely because the detection failure is what prevented anyone from noticing there was something worth recording. For most people alive today, this entire genetic quirk is invisible and irrelevant to daily life. Nobody encounters hydrogen cyanide in ordinary circumstances, and the trait matters almost exclusively in narrow professional and industrial contexts: toxicology labs, certain manufacturing industries, mining operations, and forensic pathology. Yet, it's a genuinely interesting piece of the puzzle that makes up ordinary human variation, a trait carried silently by billions of people, almost none of whom will ever have an occasion to learn whether they have it or not, since the one situation that would reveal it is by design a situation most people are fortunate enough never to encounter.
There's a temptation telling a story like this to wrap it up with something tidy, a clean moral about science triumphing over superstition, or clever chemists outsmarting careless poisoners, but that's not quite what this story is about. This is a story about how much of what any of us assumes is universal, a smell everyone can detect, a fact everyone can verify, a clue that gives away the whole plot, turns out, on closer inspection, to be true only for some fraction of the room. Forensic history didn't fail because early investigators were foolish. It struggled, in part, because it was built on the quiet assumption that human perception is a standardized instrument, when it never was. Every coroner who smelled nothing unusual at a scene where cyanide was later confirmed wasn't necessarily negligent. They may simply have been born without the specific genetic wiring for that one specific smell, a piece of themselves they had no way of knowing was missing, doing an entirely reasonable job with entirely incomplete information. That's the pattern worth sitting with, long after the details of hydrogen cyanide in cytochrome C oxidase fade from memory. History isn't strange because the people living through it were careless, foolish, or different from us in some fundamental way. History is strange because people were almost exactly like us, reasonably intelligent, reasonably careful, working with whatever information their senses and their era happened to hand them, and just as capable as anyone alive today of confidently trusting a signal that, for a significant number of them, was never actually there at all. If you want more videos like this, subscribe.